Good evening, ma'am.
Good evening, sir.
Good evening.
Sir, you can start.
Yes, ma'am. Thank you. Thank you so much. Good evening, my dear students. Once again, you all are welcome to the biological classes. Today, what we are going to discuss, the restriction, endonucleases, and their types. First of all, you should know what is my restriction endonucleases. Last class already we discussed what are the exact mechanism of the restriction endonucleases and what is my exonucleases. Okay, all those things already we discussed, isn't it? Today, who discovered the restriction endonucleases and what are the types of endonucleases? That things we are going to discuss. The restriction endonucleases were discovered by Orber, Northern, and Smith. Okay, the scientist names. Sometimes they may ask the question, restriction endonucleases. Nucleases is nothing but the enzyme which act upon only in nucleic acid only. That is very, very important. Okay, so the restriction endonucleases were discovered by, who is that? Orber, Northern, and Smith. The foundation of recombinant DNA, okay, so simply called as rDNA, were laid by the discovery of restriction enzymes. This is the very, very important for the restriction enzymes. These enzymes are present in many bacteria where they function as a part of their defense mechanisms. Already we discussed that things also. The restriction enzyme only produced by the, which one and bacteria only. Okay. Good evening, Dhanalakshmi.
Good evening, sir.
Good evening, Paul. Why are you so sad? Your voice is very less. Why?
Not sad, sir.
Okay. Anyhow.
Yes.
Okay. Last class, what we had discussed, restriction endonucleases, which was discovered by the name of the scientists Orber, Northern, and Smith. And the foundation of recombinant DNA is simply called as rDNA, were laid by the discovery of restriction enzymes. This is very, very important. And the next point is these enzymes are present in many bacteria. So already we discussed restriction enzymes secreted by only bacteria only, where they function as a part of their defense mechanisms called restriction enzymes. What is the main role of the enzymes and restriction enzyme within the bacteria? Means they are all act as a defense mechanisms. Okay, and then cut DNA of any source into short pieces. When you are using this enzymes act upon the DNA molecule, already you know a little bit, it made a cut. It is as short pieces in a sequence of specific manner, in contrast to most other enzymatic, chemical, or physical method which break DNA randomly. Okay, so this is the main mechanism, main functions or vital role of the restriction enzymes on the DNA molecule compared to the other substances, enzymatic and chemical and physical, whatever it may be. That restriction endonuclease is very, very important part. Okay, that is very, very important, my dear students. These defense enzyme, 100 bases have been discovered, 100 bases. This enzyme containing how many base pair is that? The restriction endonucleases is nothing but the enzymes containing how many nucleotide base pair? They may ask the question, 100 bases. To protect the host bacterial DNA from foreign organisms, that is called as primarily infective phages. Primarily infective phages, they told. What does that mean? So when the foreign particle enter into the host DNA, automatically what happens? The defense mechanism is activated by the enzyme is called as restriction endonucleases. The restriction endonucleases act upon the invading foreign particle. They cut into small pieces, otherwise called as fragments, small pieces. Okay, so that is a very, very important part. However, they are present only in the cell that also have companion enzyme that methylated the host DNA. This is also sometimes they may ask the questions. Okay, how it will be act on the DNA molecules. they act upon with the help of the companion enzyme. That companion means addition with the support. The enzyme methylated already you know about CH3, I think, isn't it? And then coarse DNA rendering and unsuitable substrate for the digestion by the restriction enzymes. So that is very, very important. The site-specific DNA methylases and the restriction enzymes always exist in pair in the bacterium. So this is the exact mechanism. What happened here inside? Once again, repeat for you. A defense enzyme is containing 100 base pairs. Okay. So protect the host bacterial DNA from the DNA from foreign organisms. Already you know about it. It is nothing but the invading. Invading means foreign, any other. Okay. However, they are present only in the cell that also have companion enzyme that methylated. Methylated means already you know about that. So addition with the component. Methylated the host DNA, rendering an unsuitable substrate for the digestion by the restriction enzyme. That is a very, very important one. The site-specific DNA methylases and restriction enzyme always exist in pair in a bacterium. So this is the important point. Next, we are going to discuss about the type of restriction enzymes, how they are classified. Okay, restriction enzyme already you know what is the main mechanism. Okay, they are classified restriction endonucleases. Endonucleases. There are three major classes of the restriction endonucleases based on the type of sequences. Recognize the nature of the cut made in the DNA and the enzyme structures. These are the 3 points, is very, very important. First one is based on the type of sequence, first point. Second one is recognize the nature of the cut made in DNA and the enzyme structure. These are the 3 points, very, very important. And 3 major classes have been reported or classified. Okay. So first one is type 1 restriction enzyme, type 2 restriction enzyme, type 3. So simply called as 1, 2, 3. Okay. There are 3 major classes of the restriction enzyme have been reported, identified. So many enzymes also there, but particularly according to these basic principle, they divide into 3. That is very, very important, my dear. See that one? So the properties, there are so many things in there. Okay. Abundant, where it present, recognizing site, and restriction and modifications, nucleus subunit structure, cofactor. It is very, very important for the activation of the enzymes. DNA cleavage requirement, enzymatic turnover, DNA translocation, site of methylations. These are all very, very important properties of the restriction enzymes. Already you know restriction enzyme, that is type 1 restriction enzyme, type 2 restriction enzyme, type 3 restriction enzyme. Already we studied 3 major classes. Abundance, where it locates more. See that? Less common than type 2. Less common than type 2. Most common. So most commonly found in so type 2 one there, type 2 restriction enzyme only mostly found in all the microorganisms. Just note it down. This is very, very important. Sometimes they may ask the questions, which type of endonuclease is present in common? Most common like that. This is the rare one. Less common, most common, rare one. 3. Whereas there is a recognizing site, which one is the correct one? You know, the cut both stand at a non-specific location, 1000 base pair away from the recognizing site. This is the point. But this is a common one. Already we saw it. This cut both stand at a specific, usually polyandromic. This is very, very important. That's why it comes under the getting types 2. It's most common. Recognizing site 4 to 8 base pair. Already we discussed. This is also very, very important. Okay. Sometimes it may be 100 base pairs. Already we studied. But type 2, 4 to 8%. Type 3, the cleavage of one stand. Cleavage of one stand. Here the both stand. Here also both stand. But in this part, one stand only 24 to 26 base pair. Downstream of the 3 days recognizing site. This is very, very important. Downstream. Okay. So which stand, which stand it will be cut. It make a cut like that. 3 dash end one. 3 dash recognizing site, not in 5 dash. But in this category, 3 dash as well as a 5 dash. Both the stand it will be. Okay, that is very, very important. Restriction and modifications. How it will be modified? Single multifunctional enzyme. The type 1 is a restriction enzyme act as a single multifunctional enzyme. Separate nucleases and methylase. This is important. Now we were studied, isn't it, before? The separate enzymes sharing a common subunit. These are another point. Okay. See, this one is very, very important. Just note it down. This point is very, very, very important one. This one is very rare only. Okay. Sometimes they ask the question, but the second one is a very important in the NEET point of view. And then the nucleus subunit structure, how it will be look like then? Heterotrimer, 3, homodimer, see, heterodimer. So these two are same, but in this category, homodimer. Dimer means 2. Heterodimer means 2 into 3, like that. Okay. Different, 2 hetero means different. Already you know different species are different. What is a nucleus subunit present in the different organisms. Homodimer means same. Okay, like this. What are the cofactors involved in this process during the restriction enzyme activities? Okay, the ATP, that is the energy recurring process. All are they need only that's Mg2. See that Mg2. So the magnesium is essential for the activation of the restriction endonucleases. Particularly, it is act as a cofactor. Then in the reaction will be complete. Otherwise, without the magnesium ion, what happens? No, without the cofactor, the restriction enzyme is not act properly. Okay, that is very, very important. See that ATP, Mg2, SAM, SAM. I will tell you what is meant by SAM. Okay, here also the type 2 also Mg2, all are divalent cations. Okay, SAM. Here ATP is not required in this process. Here also no need of a SAM and ATP. Here ATP, magnesium and SAM. Okay, start this one and you cut one by one. Okay, see here ATP. Next one, the ATP SM is cut. Next one is ATP only cut, like that. DNA cleavage requirement, 2 recognizing site in any orientation. This is very, very important. 2 recognizing site is very important. Here, single recognized site. Here also say 2 recognized site in a head-to-head orientations. This is also another. Enzymatic turnover, no, it will not return. Here, it will return to the another process. Another cycle is also possible. Here also. Okay. DNA translocation, first one is yes, second, both are not known. And site of methylation at recognition site, at recognition site, at recognition. So these all are very, very important. Recognized sites all are same, common. Okay. This is the exact property of the restriction endonucleases, type 1, type 2, type 3. Okay. Here you must note it down. Recognizing site, where is it? Okay. So methylations, where there is a modification takes place. Methylations, how it will be. Cofactor. Okay. And then site of methylation and DNA translocation sometimes. Turnover, sorry, enzymatic turnover. Okay. It is possible or not. See that. First one. Type 1 restriction enzyme, we are going to discuss deeply. These enzymes have both restriction and modification activities. The type 1 restriction enzymes, it has both restriction and modification activities. Cleavage occurs approximately 1000 base pair away from the recognition site. This is very, very important. First one, just take a note, that is very, very important. Restriction 1. So the approximately 1000, what is that? Base pair away from the recognized site. They require ATP and magnesium for activity. SAM also, okay, isn't it? So these are the very, very important points. The next, type 2 restriction enzymes. The restriction and the modification are mediated by separate enzymes so that it is possible to cleave DNA in the absence of modifications. Okay, so this is very, very important. The restriction and the modification are mediated by separate enzymes. So the mediation is also very, very essential for the type 2 restriction enzyme. Then only it cleave the DNA molecule. So suppose if it is absent, what happens? The modification it will be occur. That is very, very important. And then the cleavage of nucleotide sequence occurs at the restriction cycle, already known. These enzymes are used to recognize the rotation all symmetrical sequences, which is often referred to as the polyandromic sequence. Already we discussed this part. Two only very, very important in all the microorganisms, particularly restriction to enzymes. But these enzymes, what is that? Where it will make a cut, so the polyandromic sequence. Okay, both the stand already this is the normal one. Normal. They are ideal as a tool in gene cloning because they cut in a predictable and consistent manner at a site within their adjacent to the recognizing sequence only. They need the magnesium ions as cofactor, ATP not needed, and require Mg only. So here, only the magnesium ion is essential, and also ATP is not there. The energy requiring process is not there. This is called as, suppose, active and passive. What is the meaning of active reaction? In passive reaction, already studied in the where the absorption of water molecule in the ascent of sap and so on, so cohesion, okay, absorption, active absorption and passive absorptions. Okay, active means A, first letter A, so that type is a require of energy. Passive, they no need of energy. That is the relationship. Likewise, here also ATP not needed, so it comes under getting passive like that. Okay, the next one, see that one. This is the E. chloride DNA molecule already know about. This is the polyandronic sequences. See that GAA TTC. So read the opposite manner is the 5' to 3's. Here the 5' to 3's is GAA TTC. See that GAA TTC. GAA TTC. GAA TTC. TTC. So when this is the place, the polyandronic genes. Okay, the cut, how it will be made? See that restriction enzymes. See that it's made like this cut. Okay, this is the cut. Cleavage of E. chloride 1 and sticky, and you will get it like this. Okay, this is also made by the restriction enzyme 2, but I have mentioned here 1. Okay, so this is the exact locations happen. This condition is called a polyandronic. The restriction enzyme cut the strand of the DNA a little away from the center of the polyandrome site, but between the same two base on the opposite side. Already been overlooked. This leaves single stranded portion at the ends. There are overhanging structures called sticky end on each strand. Already we discussed what is by sticky end. So this part is called a sticky end. Okay. When you are making a cut, already yesterday we saw it, isn't it? Yesterday we saw it. See that one? So this is the part DNA. Already we discussed. This is the part. Okay. This one. Okay. This is a 5'. Same one already discussed. 5' and a 3'. Here also 5'. This one is called as 3'. Okay. When the cut made by the restriction enzyme 2, the cut will be like this, isn't it? So this is a structure like this. The structure will be like this, isn't it? This is the site, exact site. So after that, what happened? You will get the molecules look like this one, isn't it? It look like a molecules. How it will be? See the structures. Yesterday we saw it. That part also same one. Okay. So like this. Okay. This is the one part, and this is another part. The first, the upper part is the last one, long one. Here there is a small one. So this is the part. It's exactly match with each other, isn't it? So likewise, you will get it. So this is the part. These two joined together, you will get this one. When the restriction enzymes activate upon it, what happens? You will get this one. So this end is called as what is known as sticky end, isn't it? This part is sticky end. This is also a sticky end. Okay, that has been given here. Okay.
So video is not visible.
Now it will come. Wait,
Yes, sir.
wait. You got it, no?
Yes, sir. Now.
Yes. That's the reason. Okay. These are named so because they form a hydrogen bond with their complementary counterparts. The stickiness of the end facilitate the action of the enzyme DNA ligases. After that, what happened? When it separated, and which one is interested genes also isolate from the different genes, and also add with the foreign DNA molecule with the help of the enzyme is called as DNA ligases. That's why they told without DNA restriction enzyme, without other enzymes, it will not complete the actions. Suppose restriction 2 enzyme is very, very important for the process. Suppose DNA ligase is not there, no, what happened? The linking process is not completed. So what will be the use of the restriction enzyme? There is no use of the restriction enzyme. That is the important point. See that. And then third one is already know about types 3 restriction enzymes. These enzymes recognize and methylated the same DNA sequences by cleave 24 to 26 base pair away. Last one we saw it 4 to 8, isn't it? Here how many base pair? 24 to 26 base pair away. The magnesium ions and the ATP are needed for the DNA cleavage. And the process of cleavage is stimulated by SAM. Already we discussed it. What is meant by SAM? Okay. Self-assembled monolayer, simply called as what is that? Self-assembled monolayer. The SAM expansion is a self-assembled monolayer. Okay, this is very, very important. SAM, ATP, magnesium. These are the three is very, very essential for the activation of restriction enzymes. The cleave only one strand. Already we discussed only one strand. Other two enzymes are they may make a cut in the two strand, but in this category, cleavage only occurs in the only one strand only. Two recognized sites in opposite orientations are necessary to breed the DNA duplex. Duplex means exact copy of the DNA molecules also. How it will be? They need which one? Opposite orientation to recognition sites. This is also a third one. Okay, third enzyme. How it will be? See, the type 1 and 3 restriction enzyme are of little practical use because they use ATP for energy. They cut DNA and predictably, and their cleavage site is 100 base pair away from the recognition site. So 1 and 3. Okay, 2 is not comes under the headings. This process, what is that? Function. Okay, don't forget, my dear, this is very, very important because the restriction enzyme is types of restriction enzyme 1 and 2 and 3. How it will be, where it will made a cut. Okay, that thing's also very, very important, my dear. Okay, don't forget. See that. So that once again, what are the types of enzymes? See that how many types of enzyme is there, and which source they obtain the enzymes, the recognized sites. What are the recognized sites names, and whether the nature is how. Whether is the nature is how. So first one already you know about. Already we discussed E. coli. Okay, Escherichia coli restriction enzyme 1. Okay, see that the source already know this comes from the bacteria. Which bacteria we are isolating the enzyme? They have given as a name. Suppose Bacillus B, Haemophilus H. Okay, influenza, Haemophilus influenza. That's why HIND means what is the first letter indicating the genus name, and IND indicates that means the second one is indicate the species name. See here, HIIFL, Haemophilus H. First next two letters. Which one? I. Influenza. See this one. Okay, and then SAW3A, Staphylococcus aureus. PVUI1, Proteus vulgaris. PVU. BAMH1, Bacillus amyloliquefaciens. Bacillus amyloliquefaciens. That's a amylo, AM. You see that second letter AM. B first letter, isn't it? Genus name and species name. This one genus name, this one species name. Because each and every microorganism are named by the binomial nomenclature, isn't it? That's why the name of the enzymes also what happened, represented the bacteria also, bacteria name. Okay, so first of all, you should note it down. E. coli 1, Escherichia coli 1. Here, recognized site have been given GAA and XCC. What is the meaning of XCC? Okay, extra. Okay, GAA and XCC. It is also having the nature sticky end. When it act upon the DNA molecule, it produces the sticky end. All are having the same process. Okay, sticky end only. And BG3, Bacillus globigi. See that? AGA TCT. This is also a sticky end. Adenine, guanine, adenine, thymine, cytosine, and thymine. Hint 3, it is very, very important, my dear students. This is very, very important. E. coli also very, very important. And one more is that BAM H1 is also very, very important. PBUL. These 4 points is very, very important. 1, 2, and also these 2. First one and third one, and the last 2. These 3, 4 is very, very important in the NEET point of view. Okay, hint 3. So hint is denoted as hemophilus. Hemophilus influenzae. This is caused by the microorganism influenza. Already you know about. Okay, see that AAG. See here, the A is comes in this side. The T is also come in this side. So compared to these 2. Okay, so a sticky end. And then Haemophilus influenza. Same one, but here 3, here the 1. See that GANT, GANTC. N is a different one, isn't it? X is also different. N is also different. GATC, sticky ends. So that another one is called Staphylococcus aureus. SAU 3A is the 3A. See that it is a subunit, subdivision is also having, isn't it? GATC. The N is not here. Okay, GATC. Next one is very, very important. PBU1, Proteus vulgaris. Proteus vulgaris. All are microorganisms. Note it down, my dear. All are microorganisms, particularly bacterial species. Vulgaris. CGA, see that one? CG. Then now, and center there is at 80. Okay. See here, CG at. Here, GANTs. Okay, likewise. Here, GAT. Okay, so likewise, CG, AT, CG. So center there is a at. It is also produce a sticky end. And BAM H1, Bacillus amyloliquefaciens. Okay, so this is also a very, very important. Here, what happened? Here, see that? GGA, TCC. GGA, TTC. Okay, TCC, sorry, a sticky end. It is also produce a sticky end. So these are the enzyme, these are the sources and recognized site, whether the nature of the stand. Okay, so go to the next one. Here also having TACU1s. This is also Thermus aquaticus. Thermus aquaticus, what is the meaning of this one? You know, thermus means high temperature, it can survive that type of bacteria. What happened? Oh, produce the enzyme. That enzyme is called as Thermus aquaticus. Thermus aquaticus. See that TCGA, it is also produce a sticky end. Another one is called as SFIL, Streptomyces fimbriatus. See that a different component is there. See the center there is a 5N is there, but both are same. See the GGCC, GGCC, but center there is a 5N is there. This is also produces sticky N. Nitrogen is also combined center. And SAIL, so simply called as SAIL, Streptomyces albus. Okay, GTCGA, so sticky end. And HIND 2, Haemophilus influenza 2. So here GTP, YPUAC. Okay, pyrimidine purine. Okay, this is called as pyrimidine purine. AC. This is a blend end. Already we discussed what is by blend end. Okay, it will not produce any separate uni-nucleated base pair. You know what about the blend end, my dear? Dhanalakshmi?
Sir, blunt end, sir.
Yeah, do you know that one?
Sir, are you asking meaning, sir?
No.
Then
What is
what's
the what is meaning by what is mean by blend end in DNA molecule? What is my sticky? What is my blend? You don't know, isn't it?
no, sir?
See, now only we saw it, Nada. So this part is what is that? Here, so this is the DNA sequence. Okay, the DNA sequence. What is that? You know already know about. Okay, this is the DNA molecule. It has 2 strand, 5 dash, 3 dash, 3 dash, 5 dash. This is the enzyme cut. Which area? You know, polydromic. Okay, polydromic. Already know about. So after that, you will receive this type of structure. Okay. This sequence is simply what is that? You know, the single sequence present in the end is called as what is that? This type of A. For example, here it may be G.
Sticky end.
Yeah, this end is called as what is that?
Sticky end.
Yeah, so then it can be easily matched, you know. So this one is also. Suppose if it is A here, you know, so what happened here? A is also here means this is a T, isn't it? Adenine and thymine. So here G is there. Okay, G is here. So what about here? How it will be joined these two? In this part, automatically, you know, variable C is there, here G is there. Here A is there, T is there. So the single base pair is present in the end of the DNA molecule is called as what is it? No, sticky end. Okay, sticky end. Okay, sticky end. This one is called a sticky end. When it combine contact close together, what happens? Automatically, it will be reproduce the exact DNA molecule, the structure. This is called as what is a sticky end. Suppose some of the bacteria, some of the DNA molecules, what happen? You know, it will undergo the cut or separation or fragmentation. During that, what happened? There is no any fragmentation. That means any sticky end is there. So simply they produce just like this one. So here there is no any sticky end in this part. Okay. In this part, there is no sticky end. So this is the sticky end. This is called as what is a blend end. Okay. Blend. BLUND. Okay. So this is the difference between the blend end and the sticky end. That things we are studying here. Just to note it down. Don't forget. Okay, that's the things. Okay, blend, BLUND. Okay. So the next one is called as HPAL, HAPL, Haemophilus parainfluenza. See that same things that GTT, AAC, blend end is also produced, and HAE3, Haemophilus. So Haemophilus aegyptius. Okay, see that double G and CC, 2G and 2C is blend. SMAL, Serrata. Okay, so Serrata mariceus. See this one. Okay, it is also blend end. How many enzymes? There are so many enzymes have been reported here. Marasescence. Okay, these are the enzymes have been reported in your book. Just and understand it. So sometimes they may ask the questions, which one, you know, a blend end, which one they produce blend end, which one is not the blend end. So sometimes it may be which one of the following is a blend end, otherwise called as add one out. Like that, they may ask the questions. Okay, that is very, very important, my dear. See that blend end also can be ligated by DNA ligase. See that this is blend end. Okay, it can be easily what happen, you know, joined together. This is the blend end. It can be joined together. It will produce a which one, you know, exactly. It's a DNA ligase with the help of the enzyme. However, whereas the ligation of the sticky end recreates the original restriction site, and any blend end can join with any other blend end and not necessarily create a restriction site. This is very, very important. This is the DNA molecule, sticky end is the blend end. See, so sticky end already we saw it. I saw it, isn't it? So here there is a extra DNA molecules. The extra nitrogen base also present in the end of the DNA strand. Here also there is a extra, isn't it? It can be easily fixed. Okay, but in this category, what happened? The blend end, what happened? It it is it can join any blend end of the other enzymes, but it is the sticky end only pair with the exact copy only. See that this is called a sticky end of E core 1, the blend end of the E core 5. Okay, see that. So GAA TTC, GAA TTC. This is called as polyunderpeptide. When they made a cut by the enzyme E core 1, you will get this structure. This is the, see the single nucleotide is there. This is the sticky end. Okay, it pair with the only complementary with the another one, another strand. This is the complementary of this one, this one. This one complementary of this one is complementary of this one, isn't it? So when it's split like this, you will get it, isn't it? I show it in simply line diagram. So here they have given as a nucleotide base pair aspect. Okay, so this one is a single base pair. At the same time, what happened? Remaining present here. Here there is a single base pair is also remaining is there. It is exactly combined, common contact close together. As a result, what happened? It will be produced dihybrid nature, otherwise called a skeletal structure. But here, what happened? The blend end is there. See that blend end. Instead of the blend end, it can bind with any another blend end also here, and another blend also here. Okay, so I will get here 4 type of sequences, isn't it? Here one. Another 2 is also joined together, X cross like that. Okay, so as a result, you will get it a different, what is a nucleotide base pairs. That is very, very important. This is the blend end, this is the sticky end. You know the difference between these two. Okay, the restriction endonucleases nomenclature. Next, we will pass on to the next one is called as, what is that? Restriction endonucleases nomenclature. What is the meaning of nomenclature? Dhanalishmi? What is my nomenclature?
Naming, sir.
The naming of the plant or animal or whatever it may be, isn't it?
Yes, sir.
How many nomenclature do you know, my dear?
Sir, means what, sir?
How many nomenclature already studied in the botany?
Sir, I know few, sir, which are there in the chapters. I know, sir.
Will you please?
Sir, for Neelakuranji, Strophilanthus kuntiana.
No, I'm asking about the nomenclature. What are the types of nomenclature I'm asking, my dear?
Sir, I don't know, sir.
What about taxonomy?
Oh, yes, sir.
What are the nomenclature already studied?
Sir, I'm not getting it, sir.
Dhanalakshmi, isn't it your name, isn't it?
Yes, sir.
How many names is there?
Sir, 2 names.
2 names. So they comes under the heading is known as?
Nomenclature.
No, what I'm asking, no. 2 names means you are having 2 names, isn't it?
Yes, sir.
It has a specific term, in biology.
Yes, sir.
What is that?
Sir, bio means living things, sir.
No, what I'm asking, no, you are not come to the point. Okay. What about binomial culture? Binomial.
Sir, it contains 2 names, sir.
That's why I'm asking, no. Binomial nomenclature. That's why. But here we will not consider as a Dhanalakshmi. We cannot be able to split the genus name, species name. So for example, I have taken for you. Okay. So binomial culture. Okay. So that is that point I ask you from you. Binomial.
Yeah.
What about trinomial?
Sir, I don't know, sir.
You don't know. Okay.
No.
So binomial, what is the concept is there? No? Binomial is also any one of the plant or animal we are calling with the help of 2 names or 2 words. Are you clear?
Yes, sir.
Okay. Trinomial means what is it? First name is the binomial means first name is called as genus name, second name is called as
Species, sir.
species. What about trinomial? Means we are calling?
Don't know, sir.
Okay. I will tell you, no need to worry. So taxonomy already studied that things also. See that one? So that is very, very important, my dear Silisa. See that there are 3 nomenclature have been reported in a book. First one is called as binomial. Who proposed binomial? Do you know? Do you know binomial? And second one is called as trinomial. Sorry. Trinomial. Okay, just you put trinomial. Okay. And then third one is called as, what is that, Third one is called as polynomial. Okay. Polynomial. Binomial means, what does that mean, So any one of the plant or animal, we may call two names. Okay. Two names. Two names. The one name is called as genus name. Another one is called as, what is that? Species name. Isn't it? This is the binomial. This was first introduced by the name of the scientist Cosbert Bayan. But the binomial nomenclature was used by the Carolus Linnaeus. Who is the father of binomial nomenclature? Means Carolus Linnaeus is the father of binomial nomenclature. Are you clear now?
Yes, sir.
Okay. What about the trinomial means? See, tri means three, isn't it? Three names. Three names, isn't it? But what they are giving here, so already you know very well about any one of the plant names, genus name, species. Example, you can tell Mangifera indica, isn't it? Mangifera, okay, Mangifera indica, isn't it? So indica. So this is a name indicate the mango, isn't it? Indica. The first name, what is that? Second name, what is that? Already know about it. First name is the genus name. Second name is called a species name. So that's why it is called as a binomial nomenclature. Okay. Trinomials, what does it mean? So the same is also there, but addition of one more name is also added here. Genus name, species name, and simply they called as subspecies name. Okay. Subspecies. Okay. Species name. So genus name is also there, and species name is also there, and another one is also added here, subspecies. Some of the plant or animal, they may give three words or three names. Such type of nomenclature is called as trinomial nomenclature. Example they have given is a cauliflower as well as the cabbage. Both are same genus name, same species name, but the subspecies will be differ. That is very, very important, my dear. This is also so many times they were asked, I think 2017 or 16. This question is that. Okay. See that Mangifera indica, this is the binomial. Okay. So don't forget. And trinomial means I give you examples. So then only it will be better for your understands. Okay. See here. Trinomial nomenclature. What is it? Cabbage. And also another one is called as, what is that? Cauliflower. So these two are examples. First one is, what is that? Cabbage. Okay. Why I am telling no? So this is very, very important. Cabbage and cauliflower. Cauli. Okay. Cauliflower. Okay. So both are comes under the headings known as Brassica oleracea. What is the genus name and species name? Both are having the same things, Brassica. Brassica oleracea. 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 So this is the genus name, this is species name. Genus name and species name. Both are same. Okay. Both are same. Okay. What about the subspecies name? This is the difference. Okay. So cabbage. Okay. Portidis. Cauliflower. And one more is the albicus. So the subspecies will be differs, but the genus name as well as the species name, both the plants are same. So here, how many names given here? No, Brassica oleracea, Portidis. Brassica oleracea, what is the albicus? Okay. So this is the difference. The subspecies will be differs. That's why here, how many names given here? No, 1, 2, 3. Genus name, species name, subspecies name. That's why it comes under the heading known as trinomial nomenclature. You got it now?
Yes, sir.
This was given by the name of the scientist called as Julian Huxley. The name of the scientist called as Julian Huxley. Okay, this is the Second point, binomial nomenclature, trinomial nomenclature. Binomial already given, two names or two words. Okay, first name is called as, what is that? A genus name, and second one is called as species name. Okay, at the same time, trinomials mean same as it is genus name, species name is same, both the plants. At the same time, subspecies will be differ. Example, cabbage and cauliflower. Cabbage, Brassica oleracea portidius. Cauliflower, Brassica oleracea albicus. Okay, likewise, it's differ. And the last one is called as polynomial, isn't it? Polynomials means already you know about more than one. This is also so many times they may ask the question more than one, more than. Okay. Okay, more than one. More than one means it has a, what is a poly, more name is there. More than two is called as polynomial. Trinomial is also called as a polynomial, but here polynomial means given examples. One of the special example has given in a book. Don't forget, my dear. Now listen here. Caryophyllum suctalis folius. Sorry, yeah. Listen here. Caryophyllum. Okay. Caryophyllum. It's Y-L-L-U-M. Caryophyllum suctalis X-A-X. I typed it wrong. That is better for you. Understand. Okay. Caryophyllum suctalis. This folius, F-O-L-I-U-S, umbellatus. Okay, U-M-B-I-L-L-E-T-I-E-S. Okay, and Caryophyllum suctalis folius umbellatus, like that. Okay, so it comes like that. Okay, here the Caryophyllum suctalis folius umbellatus. Umbellatus is nothing but the it is not a flower. Folius means folius, F-O-L-I-E-S. Okay. Maximum it's present the rock, suctalis, caryophyllum. Caryophyllum means already you know available. It's a vegetative reproduction also done. Okay. So all the plant characters is also mentioned as a name or called as a name is called as polynomials. Once again, repeat for you. All the plant character expressed as a name is called as polynomial. Once again, repeat for you. Binomial, you know, trinomial, you know. Polynomial means entire characters. What are the characters? What are the inflorescence is there? Where it will grow? All the habitat growth or characters or inflorescences, all the things mentioned in their name is called as polynomials. Okay. Now you got it, that point, my dear?
Yes, sir.
Okay. This is very, very important. Caryophyllum, suctalis folius, umbellatus, coryamp, that finish it up to the level coryamp. C-O-R-Y-M-P. Okay. So like that it will finish. This is the example. Sometimes they may ask the polynomial nomenclature. Which one is an example? Like that they may ask the question. This is very, very important. And trinomial is very, very important. Cabbage and cauliflower. And then binomial nomenclature, they ask the question. Binomial nomenclature was introduced by the scientist Caspar-Boyan. Don't forget. So many times they were asked. I asked the question in so many schools. They are all answered. Binomial nomenclature was introduced by the name of the scientist Carolus Linnaeus. It is not Carolus Linnaeus, Caspar-Boyan only, who used the binomial nomenclature in his book Species Plantarum. Okay, that is called as, what is it? You know, Carolus Linnaeus. Don't forget, my dear. Okay.
Okay, sir.
Okay, dear. Taken. To the end, I think, we will finish it in properly. See this. That's why I told you. Okay. Right. So nomenclature, isn't it? The restriction endonucleases are named according to their organisms. That's why already we saw it. Hind3 means, what is that? Hemophilia influenza. D3. Okay. So Hindi3. See they here. Okay. Organism in which they were discovered using a system of letters and numbers. For example, okay, there is a spelling mistake. Hind3 pronounced Hindi3. Okay. was discovered in Haemophilus influenza. Haemophilus influenza, first three letters. H denoted as Haemophilus, influenza IN. Okay, and the strain D, that's why they have given us a D. Hindi 3 means Hind, H-I-N-D. Okay, H denoted as Haemophilus, and IN denoted as influenza, first two letters, and D denoted as a strain of D. That is very, very important. The Roman numerals are used to identify specific enzyme from bacteria that contain multiple restriction enzymes. Already you know there are 1 and 2 and 3. So many enzymes also there. That's why they have given is that this Roman letter is denoted as the, which one you know? The specific enzyme. Enzyme 3, enzyme 1, enzyme 2. Like that. From bacteria that contain multiple restriction enzymes, indicating the order in which restriction enzyme were discovered in a particular strain. So this is very, very important, my dear. So restriction endonucleases nomenclature. Okay. See that one. Derivation of the E. coli name, the abbreviation E. coli, isn't it? So only one have been taken an example. The meaning, the E denotes Escherichia. CO denoted as coli. R denoted as RY13 strain. This is the strain. This is genus name. This is the species name. Already you know about binomial. Okay. RY13. See, R is you know about restriction enzyme. Okay. That is isolated from the strain name. Okay. The I first identified order of identification. Identifications. The first one, E. coli 1. E. coli 1. This is the first one. Suppose if it is 2, second identify. Suppose if it is 3, third identifications. Like that, they have given. Don't forget, this is very, very important, my dear student. E. coli 1. E. coli is simply called as E. coli for your identification or a remembrance at the time of examination point of view. E denotes E. coli, co denotes E. coli, R denotes which one you know? The strain name. Sometimes they may ask the question, which one is R? I already know is nothing but the Roman letter. Okay, so whether it's the first or second or third depends upon their identification, order of identification. This is the important things. What are the next one? They are names using the first letter of the genus name, the first 2 letters of the species name, and the first letter of the strains from the which the enzyme was isolated. For example, E. coli come from the Escherichia coli RY13. In E. coli, the letter R is derived from the name of the strain. Roman number following the names indicate the order in which the enzyme were isolated from the strain of the bacteria. Already we saw it in the tabulation aspects. At the present, about 230 types of restriction enzymes have been identified. 2 restriction endonucleases are isolated from the 900 bacterial strains. That is very, very important. Restriction 2, how many bacteria are isolated? So 900 bacterial strains have been isolated. Okay, that's why it is present in the most of the bacteria. The first restriction endonucleases was HIND2. Don't forget, my dear, this is very, 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 very important. HIND2, human influenza, HIND2. Okay, you don't forget. The first restriction endonucleases, HIND2, which always cut DNA molecule at a particular point by recognizing a specific sequence of the 6 base pair. 6 base pair. That is very, very important. This specific base sequence is known as the recognition sequence of HIND2. HIND2 means, don't forget, it is the first enzyme, restriction endonucleases discovered by the scientist. First one. The name is called as HIND2. How many base pair is there? No, 6 base pair is there. And also this specific base sequence is known as the recognition sequence of the HIND2. This is the important point, my dear. A popular enzyme used in the genetic engineering. Okay, that things we'll discuss. Just I give you an intro only, then we will discuss in the next class. Okay, reverse transcriptase already we studied. TAC, thermoaquaticus, DNA polymerase, highly thermal, high temperature to act upon it. Alkaline phosphatase, terminal transferase. These are the 4 enzymes were used in the genetic engineering. Popular enzyme were used as a tools in genetic engineering technology. Okay, so we'll see the remaining things on the next classes. I will summarize all those things in short period. Okay, see that one. What are the things already we discussed? First of all, you should note it down. So who discovered the restriction endonucleases? This was first introduced by the name of the scientists Orban, Northern, and Smith. Okay, this is also recombinant DNA molecules. Already you know very about how it will be cut. You know, the restriction enzymes also cut in any directions. Any direction, particularly the restriction site only it may be cut. That things also already we discussed, isn't it? And also, how it will be acted in the invading microorganism, foreign particles. When it enter into the foreign particle inside host DNA, it can be easily act upon it. As a result, what happens? It may be converted into a simple fragment structure. Which one, you know, the foreign particle is split into simple structure. That things also already know a little bit with the help of the methylated enzymes. And what are the types of restriction enzymes already discussed according to the structure and nature and also sequence? They may be divided into 3 types. One is called as types of 1 restriction enzyme, type 2 restriction enzyme, type 3 restriction enzymes. Okay. So here already we discussed what are the properties of the restriction enzyme 1 and 2 and 3. Particularly type 2 is very, very important. Likewise, HindIII is the one of the important one. Okay, type 2 restriction enzyme. Most of the enzymes mostly present in the bacteria only. Okay, how many bacteria means 900 bacteria. HindIII is isolated. Restriction 1 already know a little bit. They require the ATP molecule as well as the magnesium ion. Okay, at the same time, how many base pair is there? 1000 base pair. This is the first one. How it will be? The enzyme have both restriction and modification activities also done by the restriction 1. Restriction 2. Okay, the DNA is absent of modifications. It need the mediated enzymes also. That things also very, very important. Polyadentromic sequences already know about that things also. And addition with the magnesium, ATP is no need for this process. Only the magnesium ion is essential. Act as a cofactor. Other things no need. And then third one already know about what is the meaning of restriction third. Okay, so the restriction third. Here, how many base pair is very, very important. 24 to 26 base pair away from the restriction 3. Cut, yeah, media cut. Okay, and one more is the SAM. Sometimes they may ask the question, what is my SAM? Self-assembled mono layer. Self-assembled mono layer. Clearly only one stand only because to recognize site in appropriate opposite orientation are necessary to breed the DNA duplex. That thing's also very, very important. Okay, so see that 1 and 3 is very, very important. How many base pair is that? 1000 base pair. 2 already when about. Okay, so these are the different sources of the enzyme, different types of enzyme, and where there is a source, how they have given as a name, whether it may be sticky end or blended end. Okay, that things also already discussed this part. And then DNA ligase is very, very important. What is my blend end? What is my sticky end? How it will be? Okay, that is also very, very important. Restriction endonuclease nomenclature, how they are giving the name for the each and every enzymes means, say, already one HindIII as given. Pronounce HindIII. First discovered in Haemophilus influenza. Haemophilus influenza, H, first letter H, and IN is the species name in Hind. D denoted as the strain D. Already we know about number Roman letter 3. Which one is the first one, second one, third one, like that they may discuss it. Example has given as a eco or 1. E. coli and RY is denoted as strain, and 1 is the Roman letter 1 is given as a order of identifications. That things also already we discussed. Okay, so this is the, and then at present about 230 types of restriction endonucleases were discovered, particularly 2 restriction endonucleases are isolated from the 900 bacterial strains. How many bacteria? There are thousands of bacteria present in the soil molecules. Okay, soil particle. From that one, they isolate the 900 bacteria separately. Then it undergoes analysis process. Then how many restriction enzymes were isolated? 900 bacteria were isolated. Restriction 2 endonucleases. Okay, the first restriction endonuclease is HindIII. Don't forget, my dear. This is very, very important. Okay, so we will see the remaining things on the next classes, my dear. Thank you. Take care. Bye. Good night. Thank you, my dear. Thank you, ma'am.
Thank you, sir.
you.